we're going to start a little bit about this. When you think of your ancestry, most of us think that we probably came from a family sort of similar to this. And then, how many of you have done genealogical research? Okay? And don't you every now and again you kind of find that you may have somebody a little more colorful in your background? Okay? Well, tonight we're not going to be talking about genealogy. We're going to be talking about genetics. And a little bit about that it warrants to have just a touch on the DNA. And that is that DNA has been used now for many years in courts of law. In fact, DNA is one of the few sciences that actually you can have a situation where, by the findings of this scientific methodology, someone could literally be sentenced to death over it. This is not a technology that is a lot of hocus pocus. It's not a lot of theoretical things. It's almost, if you want it, probably the best way is to compare it to a map. The map, the points on the map are either there or they're not, and they do change over generations. Okay, but that's the reason why DNA is used in courts of law, is because it's quite exact. A little bit more about DNA. The human mitochondrion is an extranuclear organelle having, of course, DNA. And let's see, all the nucleotide positions and coding loci are known, meaning that we've been able to map those scientifically so we know where they're at. Okay? Now there are different segments, and we'll get into that I'm not going to get into a big detail about DNA because that's not what we're here for. But there's a couple of things that you need to know. One of them is that mitochondrial DNA is DNA that is passed from the maternal side, the maternal line. And in fact, this explains this a little bit better, is because this DNA is uniquely maternally inherited, and unlike nuclear DNA, does not recombine. All changes in mtDNA is mitochondrial DNA, which is, again, DNA from mother. Sequence are the result of accumulated mutations inherited from mother to daughter. In addition, mitochondrial DNA mutates an order of magnitude faster than does nuclear DNA, which makes it better for analysis at shallow time depths. This is very important, shallow time depths. When people are talking about courts of law, they're not talking about people who have been dead for 1,000 years. Okay? And so that's a little bit of a background as far as DNA. And now I need to just very quickly tell you a little bit about the present day theory of where we all came from. Okay, this is from Nature Journal, 1987 article, and essentially science has come down on this side. They tested DNA and found that because of how much humans have changed, and then how fast they see the change happening today, it would mean that it would have taken about 200,000 years for all humans to have diverged that much, okay, from the original DNA testing that was done. And so scientists call this woman, they call her either mitochondrial Eve or African Eve because of the predominant theory of the peopling of the earth. And so this is a little bit about that. All mitochondrial DNA stem from one woman postulated to have lived about 200,000 years ago, probably in Africa. Now, in order to, that stemmed from some preliminary research. Well, in order to get more accurate results from the research, what needs to be done is that they need to take DNA from further back and then see if that still holds. And so they actually exhumed the body of Tsar Nicholas II of Russia. Now they know exactly who his progenitors are through the maternal side from that, and we're able to trace them back to his present day living ancestors. And when they did that, they found something quite astonishing. They found that the rate of mutation, now when I say mutation, I'm not talking about that he was turning into another species, okay? What I'm talking about is that there is a hypervariable area, and there's more of an area that is more what they call the control region. And when they look at the hypervariable region, that's what they typically are looking at when they're doing DNA studies. And they found that the amount of change happening between he and his current day ancestors was 26 times faster mutation rate than what they had anticipated based on the preliminary studies, which led to, in Science, the journal Science, 1998, in an article called Calibrating the Mitochondrial Clock, said researchers have calculated that mitochondrial Eve, the woman whose mitochondrial DNA was ancestral to that of all living people, lived about 200,000 years ago in Africa, using this new clock, this new calibration, she would be a mere 6,000 years old. Brothers and sisters, how close is that to what we understand from the scriptures? This is DNA showing at least the preponderance of a Eve that lived 6,000 years ago. But then I love this next sentence here. No one thinks that's the case. Okay? Now what just happened here? You have DNA evidence and you have theory. Okay? Well, I'm going to tell you a little bit more about that here in just a second, but you have data and theory, and sometimes those things don't match up. Now, this is a second witness. This is from Trends in Ecology and Evolution, 1997. They said the hypothetical descent of mankind from mitochondrial Eve has been much debated. And then they have the 200,000 years figure again. This research team sequenced 610 base pairs of 357 individuals from 134 independent mitochondrial DNA lineages and found that such a high mutation rate would indicate that Eve lived, what? About 6,500 years ago. And again, caveat, a figure clearly incompatible with current theories on human origins. So what happened when they had these twice now? They had two witnesses of DNA showing that based on the mutation rate, that Eve would have been 6,000 years ago, right? Well, this is a couple of years later in Scientific American, August 1999 issue, that says the mystery of human origins is far from solved, but because DNA may not be as diagnostic as it once seemed. Why is it not as diagnostic as it once seemed? Because it put Eve at 6,000 years ago, not 200,000 years ago. Okay? So he says, we're back to the bones. See, it's really good that there are things coming from the fossil side. The DNA studies can just take the back seat. Now, can you see what happened? Data did not match the theory. So which was thrown out? They threw out the data. Okay? Is that good science? No, it's not a very good idea also, because we're a little bit later tonight in this presentation, you'll see that this comes up again. Okay? When theory doesn't meet data, you have to go with the data as long as it's good data, okay? And sometimes you have to throw out the theory. Now, what we're going to talk about tonight specifically are the American Indians. And this DNA controversy actually started back in 1990. You see, there was this law that was passed because the academic world would take remains of different Native Americans that they would find, and they would take them, study them, and so forth, and then they would lock them away someplace in storage and not give them back to the Indians. Well, the Indians, to them, this is sacred remains. They really want to have their remains back, and they didn't have any way to make the academic community give them back the remains. So they were able to get this law passed called the Native American Graves Protection and Repatriation Act. Now, what happened was that they said, we have a way, of testing the DNA of the present-day Indians, the present-day Native Americans, and their remains, and so we can match them up. And so we can put them into the right things, because you see, one of the problems is that in a couple of instances they had several different tribes claiming the same remains. And so they needed to have a way of being able to differentiate and make sure that the remains are going back to the proper tribe. Well, so they set out on about a 6 to 7 years quest and began doing DNA testing throughout the Americas. They tested Indians from North America, all the way from the Aleuts and up in Alaska, all the way through North, Central, and South America to the tip of South America, and did DNA sequencing of all of these different groups. Now again, remember that the mitochondrial DNA is passed from mothers to daughters to daughters. Okay? Now, this is important to understand, because this is what our children are all being taught today in high school and in our universities. And that is that, now I call this a pseudo-theory for a reason. I know it's a kind of a new word. Okay. But the reason why I call this a pseudo-theory is because it's a theory that is taught today as an absolute established fact without empirical evidence to back it up. Now, there is some evidence, but we're going to show you tonight that there's evidence that goes against this theory. But this is the general theory, that Eve began about 200,000 years ago in Africa, moved up through Asia, over the Bering Strait during an ice age, okay? And after the ice age, then they moved into North and ultimately into Central and South America. And that is what we have all been taught and told. That's how the people of the Americas came forth. Is that correct? Okay, well, when they did this research, what they found actually is that most of it, in fact, virtually all of it, supported that idea of the Bering Strait. They found out that they could take, now there are what they call supergroups, then there are groups that are subdivided below that are called haplogroups. Now a haplogroup also can be subdivided again into clade, okay? But right now we're just going to talk about the haplogroups. What they found is that in grouping the Native Americans from North and South America, that in grouping those together, that they could actually separate those into one of 4 primary haplogroups. And they, being the first to do the DNA studies, where they started with A, B, C, and D. So again, while widespread, the geographic distribution of the 4 haplogroups is markedly non-random. Haplogroup A is primarily up here in the Aleuts. Haplogroup B is found primarily in the American Southwest. And haplogroup C and D are found throughout North and South America. Now, these are not exclusive. You'll find parts and pieces in different areas. But these are the primary concentrations of these. And this is where the controversy began. All of these groups can today be linked directly with people from Asia, thereby supporting the Bering Strait idea. So, because they're all found over in Asia, and Lehi came from Jerusalem, now, a couple of things that you need to know, and that is that there are, of the 3 primary races on the earth, they're quite easily distinguished from one another using DNA. In other words, it's quite easy to tell an Asian from a Caucasian, which in the scientific terms of DNA, they call them European. So that would be European-style DNA, Asian-style DNA, or Negroid DNA, okay? So it's quite easy to tell. Well, Lehi left Jerusalem. He would have been of the European variety, okay, of bloodlines. And they didn't find it. The initial studies came back that everything that was found was Asian DNA. Well, unfortunately, that kind of started a firestorm of things. There was a molecular biologist down in Australia. He was a bishop of the church. He saw what was happening with this and became very concerned. Now, he had some other issues with the veracity of the Bible and so forth as well. But his name was Simon Southerton, and he ended up leaving the church, wrote a book about it. That book went out. Several other LDS scientists have gotten a hold of the book. Many of them have been excommunicated or have left the church. It has become somewhat of an issue, but it really didn't become a huge issue until this little group. They heard about some of these LDS scientists who are leaving the church, and they decided, you know what, let's go out and interview them. And so that's what they did. They went out, brought a camera crew, and put together this documentary, again called DNA versus the Book of Mormon. Now, this came out in 2002, the beginning of 2003. Let me just kind of give you a couple of quick ideas. This came out, like I said, in the beginning of 2003. Since that time, they actually told some colleagues of mine that they have been averaging between $3,000 and $8,000 per day in sales of this. Now, bear in mind, though, that that's about a 48-minute long video, and it's available for free on their website. How many of you have heard of YouTube? YouTube? Okay, you know what YouTube is? You can watch the whole thing for free on YouTube. But yet they're still having these kind of sales. Who is buying this? Anybody want to take a guess? Basically, every other Christian religion out there, because for the first time they have something that we have no answers for. And see, the thing about truth is if you find something that's true in science, is it ever going to not match up with truth in true religion? No. All truth at some point in time can be put into one whole, right? Okay? So if it's true physically, it also has to be true spiritually, and so forth. Well, situation is that the Christian denominations are buying these things up. Now, I can tell you that the entire city of Springville, about 2 months ago, almost every home was delivered that. I can tell you there's at least 4 other communities that I am aware of just here in Utah County that have had this video delivered to the homes. In the Bible Belt, it's worse. In Salt Lake, it's worse. Okay, missionaries are getting hit with this a lot. I was talking to, believe me, I've talked to several parents, and they have expressed very much concern because their missionaries are getting hit with this, and what's the answer to it? Now, why don't we find European DNA in the Americas? Now, the conclusion from this is very simple. The story of the Book of Mormon is told for the specific purpose of presenting another testament of Jesus Christ. The mission of the Church of Jesus Christ of Latter-day Saints is to convert people to believe in this additional testament of Jesus Christ that took place in the Americas. The archaeological, linguistic, and now this new DNA evidence confirm that the Book of Mormon and therefore this other testament of Jesus Christ are not historically true. If the story is fiction, then so are the characters, and so is its crowning event. This means the mission of the LDS Church is a fictitious account of Jesus Christ. Now, in this, one of the most powerful points was that they had an interview with President Hinckley in January of 2002 during the Winter Olympics. There was a German reporter that was there and asked President Hinckley this question in very heavy German accent. He says, "What will be your position when DNA analysis will show that in the history never have been an immigration from Israel to North America? It could be that the scientists will find out," and then President Hinckley kind of cuts in, he says, "It hasn't happened. That hasn't been determined yet. All I can say is that's speculative." No one really knows the answer to that. Not at this point. want you to remember this, not at this point, because brothers and sisters, and back in 2002, this is a prophet of God speaking. And he nailed it. The answers to this DNA question weren't out yet. Unfortunately for these folks, they were a little premature at jumping to some conclusions on preliminary scientific data rather than waiting for all of it to come in. Because what you're going to see tonight is the rest of the story. Okay. All right. Now, in the meantime, as the Americans, as we were doing the DNA testing on the Native Americans over here, the Europeans were also doing testing over in Europe. And it came down to this. Now, everybody can read this? No, just kidding. Just take a look at this map. Okay, this is from Science Journal. This is that map bigger so that you can see it. They found, now the Europeans kind of started from the other end of the alphabet, and they found that there was this group called haplogroup X, which were found in Finland, Italy, and Israel. They were actually confirmed there, as well as in Turkey, Bulgaria, and Spain. This is European type DNA. They had a conference. The European investigators and the American investigators got together at this conference, and they got comparing notes. And they go, whoa, there's something interesting going on here. Something that we didn't see over in the Americas when we were doing it, but now we can see it because you have it over here in Europe. And they found out that there was some similarities between the European DNA, some of the European DNA, and the DNA that they really didn't know where to quite categorize it in the Americas. Now, some of this next portion is going to be chronological, so you can kind of see how the events unfolded. But this is essentially, this is from Science, 1998. Anthropologists have long assumed that the first Americans who crossed into North America by way of the Bering Strait were Asians. Okay? But recently they found surprising features on a handful of ancient American skeletons that resemble Europeans rather than Asians. The new data from a genetic marker called lineage X suggests a definite, if ancient, now if ancient is more of a question, okay, because there could be European DNA that was a result of admixture from post-Columbus and post-Vikings, okay, as they came. So they needed to verify if this is ancient and goes before Columbus and the Vikings. So they needed to find if there's a link between Eurasians and Native Americans. Now a new genetic study may link Native Americans and people of Europe and the Middle East. This is from Science, again, 1998. This is haplogroup X was different. They analyzed blood samples from Native American, European, and Asian populations and reviewed all of the published studies. I love this. We fully expected to find it, that is haplogroup X, in Asia, like the 4 other Native American markers, says Brown. But to their surprise, however, haplogroup X was only confirmed in the genes of a smattering of living people in Italians, Finns, and Israelis. Now, what does this mean? Okay. Curiously, haplogroup X is found in modern populations of Europe and southwestern Asia, but not in those of central Siberia, which is now regarded as the source for the peopling of the New World. See, how did this European DNA get into the Americas if it didn't come through Asia like it was supposed to have done? Okay, this is from American Journal of Physical Anthropology, 1999. Now, they go on to say, haplogroup X has now been reported in contemporary members of 7 specific, unrelated language families. Now, why is that important? Because you see, if they're related genetically, but they're not related linguistically, that makes them more ancient. Okay, because they've had time for their languages to diverge, but their genetics are still the same. Okay, or not exactly the same, but they haven't diverged as much as the language. Okay, which are distributed throughout the geographic regions of the Canadian subarctic Great Lakes region, and this confirms its legitimacy as a founding haplogroup. So now what we have, brothers and sisters, is we have A, B, C, and D, but then we also have this X in here, and it didn't come from Asia. Now, These guys also know about this, and they have two reasons why this DNA cannot be Book of Mormon DNA. Two reasons. Number one is because finally they did find some of this X DNA in Asia. And they trot out this particular journal article about that. Unfortunately, however, they didn't read it as thoroughly as they should. They may have read the abstract, because when you get into the article itself, which we're going to go into in just a second, it addresses that. The second reason is because they found that this DNA is supposed to be too old. In other words, predating the Book of Mormon. But we're going to address that as well. This is from the U.S. News and World Report. Now, this is not one that I would typically use in a scientific situation, but they are quoting some, or not quoting, but they're relating to some other journal articles, so I just love the quote, so I decided to include it. It's called Wallace. Now, he was one of the primary investigators of the American DNA. He says, his newest and most surprising discovery is a set of genetic markers found only in the Ojibwa and other tribes living near the Great Lakes. The markers are not found in any other Native Americans or in Asia. We just don't know how it got there, he says, but it's clearly related to the European population. The simple answer would be that the DNA arrived with European colonists, but the strain is different enough from the existing European lineage that it must have left the Old World long before Columbus. This puts European DNA in the Americas before Europeans were supposed to have been here. Okay? But to confirm this, American Journal of Physical Anthropology, 2002. Now, this is towards the end of 2002. Remember, when did this come out? Beginning of 2003. The last research and so forth was in 2002. Then they had to do their editing and so forth, and so it could come out at the beginning of 2003. In this article, he says, we identified a prehistoric individual, radiocarbon dated to 1340 plus or minus years before present, that is a member of haplogroup X, found near the Columbia River in Vantage, Washington. He goes on and says, this verifies a prehistoric presence of haplogroup X in North America. They said the most convincing evidence that haplogroup X is not the result of Viking or even more recent European admixture would be its presence in ancient Native Americans. We confirmed the presence of haplogroup X in one prehistoric sample. Okay? To the best of our knowledge, this is the first evidence of haplogroup X in prehistoric America to be confirmed. And this verifies a prehistoric presence of haplogroup X in North America. Second witness. Evolutionary Anthropology, 2003 article. There is no way that these guys would have known about this. Okay? It says haplogroup X has been found in two individuals from a 700-year-old cemetery in west central Illinois. That predates Columbus, again, and the Vikings, right? Sequences are virtually identical to those of modern Algonquins from the Great Lakes region who are confirmed to be members of haplogroup X. The Algonquins from the Great Lakes region, okay, they have this X DNA. So I wanted to find out, well, who are the Algonquin Indians? What tribe are they? Well, actually, they're not a tribe. They're a language group, okay? Their subgroups are tribes. And these are some of the tribes. You have the Blackfeet, the Cheyenne, the Lenape, the Delaware. What I want you to pay attention to specifically is the Sac and the Fox and the Ojibwe Indians, because we're going to get back to that in a few minutes. So just kind of tuck that away for a second. This is from American Journal of Human Genetics, 2001 article. Haplogroup X is remarkable in that it has not been found in Asians. See, in 2001 it hadn't been found, suggesting that it may have come to the Americas via a Eurasian migration. The virtual absence of haplogroup X in Eastern and Northern Asia raises the possibility that some American Indian founders were of European ancestry. Okay, yeah. Okay. In that case, as it has been proposed, haplogroup X was brought to America by the eastward migration of an ancestral white population. So see, they still can't get away from this idea that they came up through Asia. But there's been no trace so far been found in the mitochondrial DNA gene pool of modern Siberian, Eastern Asian populations. So they're going, well, we're not sure how they did it, but somehow they went from here all the way through Asia and didn't leave any trace. Okay? Okay, you see that? All right. So then this is the one that they came out. This is a 2001 article. It says, to extend the survey of Asian mitochondrial DNAs for the presence of haplogroup X, we screened mitochondrial DNAs of total 790 individuals for the markers that define this particular lineage, X, the X lineage. Haplogroup X mitochondrial DNAs were detected only in altanes at a frequency of 3.5%. Altanes are in Mongolia, which then would lend credence to the idea that if they found it in altanes, then they could have gone from there over. Okay, this is what they thought was the nail in the coffin to the Book of Mormon. They didn't read. See, that's one thing about researchers. You find that most researchers are kind of lazy. They read enough to get what they think, and then that's it. This is what it said in the rest of the article. Okay. Same article. It should also be noted that none of the Altean X mitochondrial DNAs harbor the 225A variant or, which is a marker for a major part of haplogroup X. However, the X mitochondrial DNAs that were detected in the Altean sample did not bear these two other variants, which are characteristic of most American Indian haplogroup X mitochondrial DNA. In other words, it didn't match. It's not the same group. They couldn't have come from the Altean group. Okay, in the Americas, because they didn't match. Then they also say that several West Eurasian Altean haplogroups, including X, were detected. Alternatively, a relatively recent gene flow mediated by women of European and West Asian ancestry could have occurred at the time of the expanding Mongolian Empire. See, rather than being ancient or more ancient, okay, which it would have had to have been if it was going to be the founding lineage of the American Indian groups. They found out that it actually is probably more likely related to Genghis Khan and his group moving into the area, and then interpopulating with the population that was there, which is clear, up at 1200 A.D. Okay, so it didn't match up. Now that's the first thing. First up, they said, well, it can't be, this DNA can't be Book of Mormon DNA because why? Because they found it in the Altanes over in Asia. Well, this just covered that. The second excuse why they said it could not be Book of Mormon DNA is because of the time frame. Now, let me tell you, when it comes down to living tissue, the longer it is allowed to just deteriorate, the harder it is to sequence the information from it. And so the older and older it gets, the less the more difficult it is to get a good solid sample. So, as you can see, you're getting back into some really old time frames here. Now, this is again, I kind of find this funny because this is a kind of a catch-22 for these for Living Hope Ministries, because here they said that the X DNA, haplogroup X, mitochondrial DNA from Europe and the Near East are found to have yielded similar coalescence time. Coalescence time, that's important. Okay, 17,000 to 30,000 years before present. Well, that obviously is long before the Book of Mormon. Lehi left Jerusalem at about what? About 600 years before Christ, right? Well, 17,000 years ago is obviously long before that. 17,000 years ago, by my calculation, is approximately 11,000 years before Adam and Eve were first on the earth. And now this is a Christian denomination, and they also believe that the Bible is literal. And so it's kind of funny that they're using something against the church that they also don't believe in. But anyway, so let's address that the dating issue here for a second, because now this is from Evolutionary Anthropology 2003. This is called Conceptions and Misconceptions of the Population Prehistory of the Americas, and in this, it still blows me away that they put this in there, but it is. It says a molecular clock first suggested by these two gentlemen in the 1960s. 1960s. Has been employed to use molecular divergence to date prehistoric events. Now, I have actually had geneticists attend this presentation, and then was invited to go to the office of one of them And we had a nice, long discussion. And he said, he says, one thing about it, Rod, he says, you got to understand that every time we see a mutation, we automatically click off 5,000 years. So if we see 2, that's 10,000. We see 3, that's 15,000. And it's not based on these other things which I showed you to begin with, where it went from 200,000 to 6,000 years, right? Well, the same kind of thing is happening here. They actually are trotting out theories that were clear back in the 1960s to do this dating. That's where this dating is coming from. In fact, this is from Annual Reviews of Genomics and Human Genetics, 2005 article. He said that he showed a model that includes a rate variation in the mitochondrial control region that gave an estimate for the age of the human mitochondrial DNA ancestor. It was half of that was obtained from a single mutation rate when that was assumed. I know that's a lot of scientific mumbo jumbo maybe, but what that's basically saying is that when they checked one individual and then checked it against the population, the individual or the population ended up being half of what the original individual person's dating was. Well, how can that be happening? Half. That's a huge difference. Okay? So he says, therefore, average age estimates of the mutation rate for human mitochondrial DNA do not reflect the true state of affairs and should be viewed as simplistic tools for phylogenetic studies. In other words, for establishing relationships, but not for establishing dating. Okay? And then finally, he says, this means that care must be taken when using mitochondrial DNA to date phylogenetic events, as the underlying assumption of neutral, clock-like evolution may not hold. Okay? Why is that? Because what they found out was they were trying to do everything with the hypervariable area, and they found out a little bit later that not just the hypervariable area do, but also the control region changed, which throws everything off. Okay, so what they're saying is, don't use DNA for dating old things. It's great for current day stuff, but not for old things. You can't use it for dating. Okay, same article says, although haplogroup X is now accepted as pre-Columbian Native American haplogroup, controversy still surrounds its origin. This is in 2003, okay? Why would there be this controversy? Okay, because it doesn't match the theory. Okay? While the other Native American haplogroups are found in Central and East Asia, haplogroup X has not until quite recently been identified that far east. This has led to the hypothesis fueled by morphometric studies of the Kennewick Man remains in Washington State and other Paleo-Indian remains that there was a prehistoric migration of Europeans to the New World. But why is this a controversial thing? Okay? Because you see there's this disconnect in archaeology. Archaeologists kind of have this assumption that ancient people didn't really know how to build things called boats. And so they had to wait for an ice age in order to cross over on land bridges. Well, it's very interesting. There are a few scientists who understand this. American Journal of Physical Anthropology, 2002. I like this guy. He suggested that early Holocene Europeans used a transatlantic route to colonize the Americas. That's what his take on this DNA was back in 2002. And this goes on a little bit further. It's a 2003 article from Journal of Scientific Exploration. Pre-Columbian contact between the Americas and travelers from other continents and islands are highly probable, but the topic is taboo, okay, to mainstream American archaeologists. Why is it taboo? Again, because this means that if people had boats, they could travel. Will travel, have boat kind of thing. Okay? But here's some of the evidence he gives. Seagoing fishermen were in the North Atlantic in the first millennium BC, and probably earlier, indicated by bones of deep-sea fish in coastal sites of northeastern America. These people knew how to fish. They were going out and getting deep-sea fish and bringing them in and eating them, leaving the bones, right? Okay? In fact, he goes on to say the probability of transoceanic contacts before Columbus is so high that one might say it is a statistical certainty. Okay? Well, let me ask you, what is that? That's a boat, right? This happens to be on a wall in It's in one of the burial chambers in the Luxor area there in the Valley of the Kings of Egypt. Okay? These people far outdate or predate Lehi and his group. And my feeling is really simply this. I believe that people have known how to build a boat ever since Noah built one. Okay? All right. Now, so that brings us to this, this is really a critical article. This is from American Journal of Human Genetics 2003, called Origin and Diffusion of Mitochondrial DNA Haplogroup X. The results of this study point to the following conclusions. First, haplogroup X variation is completely captured by two ancient clades. Now remember, clade is when you have a haplogroup and you subdivide the haplogroup. So we have haplogroup X. We're subdividing that into two clades. They call it X1 and X2. X1 is largely restricted to North and East Africa, whereas X2 is spread widely throughout Western Eurasia. Now where is Western Eurasia? Okay, the Levant area or Jerusalem, the Holy Land, let's put it that way. It wouldn't be Jerusalem specific. Okay? Second, it is apparent that the Native American haplogroup X mitochondrial DNAs derive from the X2. In other words, not the ones that are from North and East Africa, which are here, but the ones that are from Western Asia, which are here on the map behind the words. Okay? Third, the few Altean and Siberian haplogroup X lineages are not related to the Native American cluster. So they come right out and say it. They're not related. So that eliminates those people. And they are more likely explained by recent gene flow from Europe or Western Asia. But the final of this is really where it gets exciting, brothers and sisters. Finally, phylogeography of the subclades of haplogroup X suggests that the Near East is the likely geographical source for the spread of subhaplogroup X2. The presence of a daughter clade in northern Native Americans testifies to the range of this population's expansion. It is notable that X2 includes the two complete Native American X sequences. So what we have, brothers and sisters, is we have European DNA, which is found right in this general area, which matches up with DNA of modern Native Americans in this area. And they have no understanding of how it got there. You see, because it couldn't have come over through Asia. Who are the Druze? Anybody know who the Druze are? Okay. It's important because this is from BMC Evolutionary Biology, 2007 article. They were testing the Saudi Arabian population. They found that the Druze sample was significantly different from all the other populations, mainly because of a high frequency of haplotypes belonging to the minority haplogroup X. See, haplogroup X isn't the majority haplogroup over there, but it is a strong minority, 27%. So who are the Druze? The Druze reside, now this is from Statistical Abstract of Israel 2006, Israel Central Bureau of Statistics. Who are the Druze? The Druze reside primarily in Syria, Lebanon, and Israel, with a smaller community in Jordan. The Israeli Druze are mostly in Galilee at 70% and around Haifa at 25%. The Golan Heights, a region controlled by Israel but claimed by Syria, is home to about 20,000 Druze. So what we have is this. Now this is a little pixelated because I took this directly from the map in the article, okay, in the journal article. Figure 5, this is the worldwide distribution of haplogroup X frequencies. The Druze, 26.7%, located in Israel. The Ojibwe Indians, 25%, in the Great Lakes area of the United States and Canada, and Orkney, which is a small group off of these islands. Okay, those are the primary regions of this haplogroup X2 group. Now, a couple of things. This is one of the oldest images that I could find of some of the Great Lakes Indians. And I think it's kind of interesting because most Indians, we tend to see them, they have the feathers and so forth. The Great Lakes Indians wore turbans. Kind of interesting. Now, this one is critical. This is 2003, Evolutionary Anthropology. Though presently thought to be common among speakers of Algonquin languages, which is, they've been, verified as that fact. Okay? This also reaches a frequency of 20% in some Algonquin populations. It is geographically widespread throughout North America. I want that to sink in for just a second. This is throughout North America. Okay? Among groups that are sharing no close historic or linguistic ties. So this brought me to the next thing. What about Central America? Okay, because where is the Book of Mormon supposed to have happened? In Central America. Okay, well, if you're trying to learn by study and also by faith, and we're told by our scholars that it happened in Central America, right? So I thought, okay, I'm going to be able to find this information. Well, what I found was a 2005 article from American Journal of Physical Anthropology. And the article is titled, "Is Haplogroup X Present in Extant, meaning present day, South American Indians?" A total of 1,159 mitochondrial DNA samples from 25 South Native American populations was surveyed for the mutation frequently found in haplogroup X. Whoa. The results indicate that haplogroup X is not present in these samples. Up to now, among extant populations, X has only been found in North America. I was going, whoa, okay, hold on. Okay, 2005 article, Hispanic American Historical Review. Ancient Mayans and contemporary Mayans, haplogroups A, B, C, D, and X. Second witness. There's no European DNA in Central America or South America. It was found in North America. So as a researcher, I had a decision I had to make. Now we have some data. We have a theory. Which am I going to throw out? See, because the data is not matching the theory. The data is saying that there's European DNA in North America, but the theory is that the Book of Mormon happened in Central America. So now I'm faced with the same situation, same decision as the scientists were faced with the DNA. And I realized that, okay, we have to get back to the roots. We're going to have to go back to the very beginning and go from that point.